Does it look like?
Yes, yes, yes.
You can see it, right?
We can your
Very
picture,
nice.
yeah, photo.
Very nice. Okay, so I'm going to shortly present the topic of my first year in the PhD, or the PhD in the group of Thomas Noll in here at Bielefeld University. If I'm going too fast, you have to tell it. And I'm also very glad that the presentation is being recorded, because I do not actually have everything written out, but I will talk you through. Okay, so the topic is called viral promoters for transgene proteins production. That's me. I was not able to connect the camera, but this picture was taken actually yesterday, and that's my office, and the background is my post.
Can you please a little bit louder? Yeah, yeah, yeah.
Sure. Are you having some technical issues, or?
Yeah, yes. You can continue, please.
Okay, thank you. So if it's going to be all right, yeah, okay, now I heard some tone. Yes, that's me, and this is the poster of my second PhD year. I had a pause in between because I got my second child. I now have two of them, and they're quite small and also need my attention, but that's not the main top point today. Okay, so the content, I will first talk about the theoretical background of the topic. Then I will present you some experimental data, and then we'll continue to the discussion. So the background, I will first explain what are the promoters. I will talk about the architecture of mammalian promoters and also their function in the cells. I will explain why the viral promoters are oftenly employed for producing some transgene proteins in the mammalian cells. And also, I will talk about the drawbacks of viral promoters. The data that I would like to present will connect to measuring of expression strength with the help of the luciferase assay. So I will briefly explain how the luciferase assay is actually functioning, and what and why and what is it able to show us. And then the results deal with one of the viral promoters, which is the cytomegalovirus promoter, known as CMV promoter. And there are two types of this promoter, namely the core promoter and also the complete sequence of the promoter. And they have some essential differences which lead to different levels of transcription. And at the end, of course, you will be able to ask the questions. And I'm very aware that the topic of my research here is very far apart from the topics which are actual for the biotechnology department at the National Aviation University. But I hope that this, how it's called in Germany, über den Tellerrand blicken. This is how, like looking on some other research topics will be still interesting to you. Okay, so let's start. So this is the biotechnology journal, and this cover picture was actually done by me, by our working group. This was the first time we were granted the possibility to do the cover of the journal issue, and the issue was introducing all possibilities of the Chinese hamster ovary cells, and this is this abbreviation CHO, which stands for Biosystem Biotechnology. I published my first review in this number of the journal, and it had the name Engineered and Natural Promoters and Chromatin Modifying Elements for Recombinant Protein Expression in the Chinese Hamster Ovary Cells. It was a very nice work, and the information that I gathered for this review will be presented in a very short form today. So just to get you familiar with the CHO cells, the Chinese hamster ovary cells are the mammalian cell lines. These are the eukaryotic cell lines, which were established in 1952 by Thomas Puck. And since then, they conquered many industrially relevant protein-producing processes. The CHO cells have two major Fähigkeiten, how do you say it? They have two major peculiarities which allow them to be employed in many different setups. The first one is that the Chinese hamster ovary cells can be easily adapted to suspension growth. This is not very common for the mammalian cells, but these CHO cells, they are able to grow like bacteria in the media. So you can grow them in the shaker flasks, which you also use in Kiev for the bacterial production, but you of course have to use another media. But still, the CHO cells are very robust. They grow very well, thus allowing a high titer of the product. From 1952, when the cell line was established, there are, of course, many different clones of it. But since then, according to the data in 2018, 85% of monoclonal antibodies are produced in the processes involving the Chinese hamster ovary cells. So if you have never heard of them and you are interested in recombinant protein production, you should maybe have a look on these cells. They are wonderful. We work with them in our working group since it was established for 10 or 15 years ago. So about the same time as the biotechnology department in the National Aviation University. The CHO cells, except for growing so well in the suspension, have another very nice Eigenschaft. I have the German words for some terms. So except for growing in suspension, the CHO cells also are able to actually fold the protein exactly as it should be for the human cells. This is not an easy thing to do, and that's the reason why bacteria cells cannot be used for many for producing the therapeutic proteins. The bacterial cells do not fold the protein correctly, and the patterns of glycosylation of the protein by the bacterial cells is also very different or not possible at all. So the CHO cells are robust. They also can be easily manipulated. You can easily imply the genetic manipulation to these cells, and they also give you a good product. So these are the three reasons why the Chinese hamster ovary cells are so overwhelming in the biotechnology and production of transgene proteins today. So in order to understand why the viral promoters are usually used for transgene production in the mammalian cells, we have to understand the difference between mammalian endogenous promoters, their architecture and function, and the viral promoters. So first, I will focus on the mammalian endogenous promoters. So here is the brief, here's the schematic picture of the DNA strand, which is showed in the gray. I hope you can see my arrow. Do you see my arrow? Can you see the arrow on the slide or not? Maybe not. Yes.
Yeah.
Okay. Thank you. Thank you very much, Mr. Sieg. I know many of you actually, and it's so interesting to see the faces to that. Okay, so the DNA strand is in gray, and here we have the gene which are interested in, and the promoter is the portion of the DNA immediately preceding the gene. So the promoter is the DNA region which acts as a transcription unit. So the promoter tells how strong the gene should be transcribed. The promoter is the region commonly known as the region where the RNA polymerase II, the transcribing complex, is binding. So the promoter, and here is the core promoter. So the core promoter is the immediately preceding portion of the DNA before the gene is being read. So before the DNA, the RNA is being produced. So you can imagine starting here, you will have your RNA product, and the promoter gives the signal how much and how exactly this RNA product should be processed, should be read. The core promoter is the very short portion, and it's immediately important portion of the DNA preceding the gene. The core promoter is the short version of the promoter. It surrounds the TSS, the transcription starting site, 50 base pairs before and 50 base pairs after the transcription starting site. The TSS is a specific nucleotide in the DNA sequence where the transcription is started. Except for this part, we have even more complex structures. Here, the promoter is also being influenced, so the core promoter is also being influenced by the distal elements. These distal elements can be spread as far as 2,000 base pairs around the transcription start sites. They also can be not only preceding the gene, because the DNA strand can loop, these distal elements can be spread everywhere. They can be also be after the gene. So to actually to actually get the whole promoter as it is in the native state in the mammalian cells, it is a very hard job. So you can imagine that this is a strand of one of the chromosomes. There are 22 chromosomes in the Chinese hamster ovary cells, and this is just one strand. And in order to get the full sequence of one mammalian endogenous promoter, you have to clone out the core promoter. So you have to actually find them and then clone them out, the whole structure. This whole structure, the promoter, the core promoter, and the enhancer elements will attract specific transcription factors, and these are the proteins that influence the level of transcription. And if we talk about the promoter strength, that means that the stronger promoter drives a stronger transcription. That means that a stronger promoter allows production of more RNA products in the, for example, 1 minute as a weak promoter. So that's the meaning behind the strong and weak promoters. Okay, this is the architecture. Now we will focus first on the core promoter. The mammalian core promoter, so these 100 base pairs surrounding the transcription starting sites, this is the picture out of my review with the whole annotation to it. Here we have, again, the DNA strand, exactly. So this is this portion, the core promoter, so we just zoom in. Here we go. We start -40 or -50. It doesn't really matter. This is the same DNA strand. Here we would have the gene of interest or the gene that is being transcribed from this promoter. Here we have the A+1, and this arrow signifies the transcription starting sites. It is usually the adenine nucleotide, and it's known as the adenine +1. So it's just a common thing. And there you place your 0, and then you are being happy. You see that the core promoter has a number of distinct elements, and all of these boxes signify, so in this DNA portion, there is a specific recognition pattern, which is usually highly conserved between the mammalian cell lines and usually also between all the species, including bacteria. So some of these regions, so these are the portions of the DNA with a very conserved nucleotide sequence. They are very small. They are like 10 to up to 10 nucleotides long. And these specific parts of the core promoter attract the factors, the proteins which bind to it, and then start the transcription. So that's how you can understand it. You should also be aware that none of the endogenous promoters have actually all of these parts. Usually, a normal endogenous promoter, mammalian core promoter, comprises out of 2 to 3 of these elements. The most known of them is probably the Tata box. The Tata box has, it's like a 7 nucleotide or the 7 base pairs sequence, which is highly conserved between archaea up to mammalian cells. And although it's mostly known and it was firstly identified, it occurs only in 10 to 20% of the mammalian promoters at all. So you can imagine how diverse mammalian promoters are. So they are long, they are very diverse. And just to add another layer of complexity to it, they are also they have not only the core promoter, but also the proximal promoter elements, which are usually referred to together as the promoter, which is under 1,000 base pairs long. And as I also told, there are also distal regulatory elements. So there are enhancers, which positively influence the transcription level. So you can imagine here we'll start the transcription on the DNA. Here you will have the gene that will be transcribed directly downstream of the promoter. And all of these small portions, the proximal promoter regions, but also these distal regulatory elements, the enhancers that enhance the transcription level, or the silencers, which decrease the level of transcription, they all together will influence how many transcripts you will get. So this portion, to get it, to imply it to the production of the transgene protein is very hard. Of course, you can find the mammalian promoters, but to clone them out and to use them for transgene production is a very hard job. And just to add another layer of complexity, the promoters in the mammalian cells, the endogenous promoters from the mammals, are of two types. There are the focused promoters, and these are the easier ones to have. They have only one transcription starting site. So all the RNA transcript that you will get, they have the same pattern, they have the same structure. They all start from the same point in the gene, in the DNA, and they will be processed the same way. So here you can imagine these are the exons, the red parts of the protein, the parts of the native protein someday when it would be translated. And in between, there is also the introns that are being deleted as the RNA is being maturated. So focused transcription is the easier part, but there's also the dispersed transcription promoter type. And unfortunately, it is even more common in the mammals. Here you have the different transcription starting sites. So the RNA is being transcribed from different starting points, and you have different RNAs flying in your culture, actually, which is not very good when you are interested in one singular protein which you would like to overexpress. So using mammalian promoters is possible for transgene production, but it's very hardish. You can also imagine that the dispersed transcription promoters allow very levered and very balanced, balanced transcription pattern. And that's not what you're interested in when you're trying to overexpress a single protein. You need a very strong transcription in order to get as many products as you can. So the mammalian promoters are very complex, and they are not very easily implied for transgene production. The viruses, on the other side, they have evolved naturally as very powerful units, actually transducing. Okay. Can you hear me and see the presentation? Maybe?
Yes, we can hear.
Yes? Okay, thank you very much for your reply. Because my connection was interrupted right now. So the viruses, they evolved naturally in the meantime of the evolution as specific agents very actively and very nicely transducing the mammalian cells. That's how the infection is actually being spread. The virus poses itself in the mammalian cells, it gives the genetic, it transduces the cell, and then it puts its genetic material to the work in the mammalian cell. And we can use this, this specific property of the viruses to actually get our gene of interest. Usually it's being done as follows. We know the sequence, the promoter sequence from the viruses, and then we can use it, we can clone it directly before the gene of interest. And because the viral promoters are usually very concise and very strong, you can get a lot of gene of interest in the end. There are three viruses or three promoters from three viruses which are usually used in the industry. These are the human cytomegalovirus immediate early promoter, the HCMV, the Ross sarcoma virus, and also the SV40, the simian vacuolating virus. The most important of them is the CMV virus and the CMV promoter. This is a very powerful unit. The CMV promoter transduces almost every cell line, so it works in almost every cell line, and it drives the highest possible transcription level. This is amazing Eigenschaft. This is the amazing, how do you say Eigenschaft? I need to just look it up. Способность auf Russisch. Wie is it in Russian? How is it in English? Способность. Can you help me?
Maybe possibility.
Yeah, the possibility. That's a nice word. That's a nice word. Thank you. Yeah. So the CMV. Thank you very much. The CMV has an incredible possibility to transduce the cells and drive the highest possible level of transcription. Okay, how do you do that? Well, you usually transduce the cells with the plasmids. This is the plasmid I've used in this work, which I'm going to present to you. So the plasmids are, of course, multiplied in the bacterial cells. You have probably heard of that before. And here is the CMV promoter. Here is the promoter, which will drive the transcription of the gene cloned directly downstream of it. And I have chosen the green vanilla luciferase because it's a reporter protein, and having it in the vector, I will be able to tell how much or how strong is my promoter actually. So the CMV promoter is the golden standard in the industry. For 30 years or about so, almost all products that you have on the market are being produced from the CMV promoter. So it's very strong. It's not regulated, but it allows a wonderful come out of the protein in the cells. So the natural CMV core is very easy, very simple. It just has three elements, which I've mentioned before. Here we have the initial starting sites, the A+1, the adenine +1. The INR is the initiation region. It's like the triad, the nucleotide triad surrounding this A+1. Here we have the Tata box. It's actually present in the natural CMV. So this virus decided itself for the Tata box, but Tata box is not explicitly needed for a transcription site start. And the CISR, this region is used for the virus, for the viral replication. So it does not play a significant role in the mammalian production. So the core promoter of the CMV is nothing special, but what about the other promoter elements? And recently, for 10 years or about so, a very powerful CMV enhancer immediately preceding the CMV core sequence was discovered, and this one looks something like that. So if you imagine the black line, this is a DNA strand, and all of these parts, so you see how dense it is, how nicely compact the sequence is. Each of these colorful parts represents nucleotide binding factors. And if you have more of them, you get the stronger transcription. So for mammalian cells, for the CHO cells, some of them are more relevant and some of them are less relevant. So you can imagine that out of all these building bricks, theoretically, a specific binding factor, and here are the names of the binding factor, of the binding factors, for example, yin yang 1 or nuclear factor 1. So these are just the proteins that would bind to the sequences in the promoter, which are marked here. So you can imagine how many transcription factors can this promoter actually attract, and that's amazing. But not all of them are used in the mammalian cells. It was shown.
Frau N.A.
Yeah.
How many time you need to make conclusion?
10 minutes? Is it too long?
Yeah, too long, please, because we expect some foreigners from United States to make report.
Yes. Okay. I'm very sorry. It's not my fault.
Okay.
Yeah.
Sorry, please.
Okay. Yes. So these are the factors which are used for the mammalian cells. So here is the overview of what are the differences between mammalian indigenous promoters and the viral promoters. And so the luciferase, the transcriptional strength can be actually measured by the luciferase assay, and it's based on the renilla luciferase signal. And the luciferase is the protein which you code in your plasmid. So then you can measure the light coming out of it, and in order to get how strong the promoter is, you can actually measure how much light you get instead of measuring how much protein you have. And to compare three promoters that I have cloned, this was the cytomegalovirus, the complete sequence, including this powerful enhancer. Then I had the empty vector as a control. This is the multiple cloning site, so there was no promoter in between. And here we had the minimal promoter before the renilla luciferase. And here's the data. This is the graphic showing the logarithmical scale. As expected, the CMV was able to drive the highest most transcription, of course, but also this the minimal promoter, the core promoter, was also quite strong. But the most interesting result is actually that the fact that the empty vector, the PMCS, was also driving quite a significant level of transcription. So if you do any transcriptional studies, you have to be aware of it, that the backbone of the vector plays a very significant role in the transcription strength. Okay, here is the conclusion. The bioluminescence-based assays can be used to measure transcription strength in transient transfection. The promoter-less vector still can drive a significant level of transcription, and the complete CMV sequence, core and enhancer, drives 358 higher transcription level as the empty vector. And here in the right, you have just the word map for the renilla luciferase. So if you deal with any types of enzymes, so you can visit this BrendaEnzymes.org, and it's a wonderful database for getting all sources of information. Okay, then I'm done. Here are the references, and thank you very much for your attention.
Thank you very much, Frau N.A.